Hello, we're glad you've joined us for this live webinar, The Use of Flow Cytometry to Identify Cancer Stem Cells in Primary Tumors and in Circulation. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. Thermo Fisher Scientific helps their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. For more information, please visit www.thermoscientific.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Bruno Sainz, PhD. Dr. Sainz is a Ramón y Cajal Researcher with co-appointments in the Department of Biochemistry at the Autónoma University of Madrid, the Department of Cancer Biology at the Instituto de Investigaciones Biomédicas Alberto Sols, CSIC-UAM, and in the Chronic Diseases and Cancer area of the Ramón y Cajal Institute for Health Research. Dr. Sainz obtained his PhD in microbiology and immunology from Tulane University and began working on cancer in 2011 after joining the Spanish National Cancer Research Center as a staff scientist. He now heads the Cancer Stem Cells and Tumor Microenvironment group, focusing on the study of cancer stem cells, CSCs, and the fibroinflammatory microenvironment in pancreatic ductal adenocarcinoma. His lab is running a combined basic and translation research program, which synergistically combines studies on the biology of mouse and human CSCs, including their in vivo microenvironment, to enhance our understanding of the regulatory machinery of CSCs. The group is using state-of-the-art in vitro and in vivo systems, along with acoustic flow cytometry, to better understand the role of CSCs in cancer. Dr. Sainz's complete bio is found on the LabRoots website. Dr. Sainz will now begin his presentation.
Thank you very much, Judy, for that presentation. I'll be very happy to get started. So I've set 3 basic goals for this webinar, as you can see. First, I want to discuss with you all how to identify cancer stem cells using flow cytometry, evaluate factors that should be considered when using flow cytometry to identify cells from tumors and from blood, and also discuss sample preparation and choosing the correct cancer stem cell markers to identify these types of cells from blood and also from tumors. So, as we all know, 90% of cancer deaths are a result of metastatic disease. And from a clinical or scientific perspective, one could say that understanding these cells or the cells that drive metastasis is absolutely fundamental. And oftentimes, the cells that drive metastasis are in fact different than the bulk tumor cells. So it's something we always need to keep in mind when we're thinking about metastatic disease. Now, I think this point was best exemplified by a study published in 2012 in the New England Journal of Medicine by Gerlinger et al., in which the authors analyzed and sequenced 9 different regions from a renal tumor, including also 3 metastases. Now, this was a very important study because they found that the tumor itself contained about 128 mutations, but only a third were present in all the regions analyzed. And interestingly, a fourth were found in only one single region. The metastasis appear to have risen or been derived from only one particular region, which is noted here in blue, region number 4. And in general, the evolution of the tumor was not linear at all, but branched like a tree. And this is something we typically see in stem cells, which also divide in a very similar manner. So in summary, this study is not only important, but it highlights the inherent heterogeneity present within tumors and the contribution that very few cells have on the evolution or basic processes of tumor evolution, such as metastasis, here reflected by the area 4, which is the only region of the tumor that actually gives rise to the metastases and secondary organs. Therefore, to turn the tide on this alarming statistic of 90% of cancer deaths are a result of metastatic disease, I think we really need to better understand the tumor at the cellular level and also develop techniques to differentiate all of the essential cellular players involved not only in the process of tumor formation, but also in the process of metastasis. Now, it's been known for the past 2 decades that cancer stem cells, or CSCs, play a very important role in metastasis. These cells represent a very rare and small subpopulation of cells within the tumor that are highly tumorigenic and have stem-like properties. To date, they've been identified in just about all solid tumors, and they are believed to be drivers of, for example, resistance to therapy, disease relapse, and most importantly, metastasis mediated by cancer stem cells that actually go into circulation, which we refer to as circulating cancer stem cells. Now, the cancer stem cell concept as a whole is not a very new concept. As early as 1855, there were reports, multiple studies reporting stem-like cells in different cancers. And we can think of Rudolf Virchow and later Julius Cohnheim as the grandfathers of the cancer stem cell concept, which they both proposed on the basis of observed similarities between fetal tissue and cancer, that cancer actually developed in the past from activation of dormant embryonic tissue remnants. We now know this is not true, but these were the first studies linking a possible link between cancer and embryonic tissue or cancer stem cells. Since then, it took about 100 years to officially isolate and identify cancer stem cells. And this was due to the efforts of John Dick and colleagues in 1994, where they, for the first time, were able to isolate and purify cancer stem cells from acute myeloid leukemia, AML. Now, this advance, or this very important discovery, was based on the availability of 2 important techniques or methodologies. On the one hand, the availability of immunocompromised mice, which is very important for the capacity to implant human cells into a small animal model, and also the advent and the availability of flow cytometry, specifically fluorescence-associated cell sorting. Both of these techniques allowed the researchers to first isolate the cells of interest and then implant those cells in nude mice to test their tumorigenic capacity. So you can see here on the right one of the experiments and figures from their paper. You can see that the authors isolated both CD34 negative and CD34 positive AML cells via flow cytometry using antibodies against CD34. And you can see very nicely that only the CD34 positive population are the cells that are tumorigenic or have the capacity to form tumors in immunocompromised mice. They went one step further and also used CD38 and isolated both CD34 positive, CD38 positive cells and CD34 positive and CD38 negative cells. And it was the latter population, the CD34 positive, CD38 negative cells, that were the tumorigenic cells. So this also showed that within the cancer stem cell population, there's a hierarchy, and sometimes multiple cell surface markers need to be used to identify these very rare and tumorigenic cells. Now, since then, cancer stem cells have been identified in just about all solid tumors, as you can see here, in liver cancer, in pancreatic cancer, and in multiple other solid organs. It's important to note that today it is well accepted that cancer stem cells are the root of the tumor and must be eliminated to ensure tumor eradication. Now, cancer stem cells maintain the tumor heterogeneity, and they promote chemoresistance and metastasis due to their stem-like properties. So if we look, and we refer to this as stem-like properties because if we look within these cells, specifically at the molecular level, we often find pathways activated that we tend to find activated and functional in stem cells. So if we look within a cancer stem cell, we can find Wnt signaling active, hedgehog signaling active, Notch signaling active, and nodal signaling active, Hippo pathways active in these cells, which are pathways that we tend to find active in cancer stem cells, and therefore we often refer to these cells as stem-like cells or cancer stem cells. In addition, the cancer stem cell resides within a very complex niche within the tumor, much like a stem cell resides within its niche. And you can see this here shown on the left, the tumor niche or the cancer stem cell niche that receives cues or information from its niche. The niche provides information that promotes a symmetric division of cancer stem cells to give rise to, for example, transient hybrid cancer cells, or which then will give rise to more differentiated tumor cells within the tumor stroma or the bulk tumor. And these cells at one point or another, either the more differentiated cells, the transient hybrid cancer cells, or even the cancer stem cell itself can go into circulation. Once in circulation, they can make their way to secondary organs, for example, the liver, and establish metastases in these organs. Now, circulation is a very difficult environment for these cells, so they have to, on one hand, survive apoptosis, and on the other hand, upregulate factors or ligands or receptors that will make them invisible to the immune system, basically promoting immune evasion. It's important to note, however, or in addition, that the relatively new concept of plasticity when we talk about the cancer stem cell concept, whereby a non-cancer stem cell, for example, can differentiate into a cancer stem cell if the cancer stem cell compartment is compromised. So this, in addition, makes this situation or this concept even more complex, and targeting, therefore, the cancer stem cell may not always be as efficient as we have previously thought, understanding that transient hybrid cells can at some time or another via plasticity take their place. Thus, since the majority of cancer patients die from metastasis, what is needed, in my opinion, are methods to detect those cells that actually cause metastasis, be it either circulating tumor cells or, in this case, and specifically for this webinar, the identification and the study of circulating cancer stem cells. In my laboratory, we study circulating tumor cells, cancer stem cells, and circulating cancer stem cells in the context of pancreatic ductal adenocarcinoma. PDAC, as I will refer to it as, is a malignant neoplasm of the exocrine pancreas. It's currently the fourth most frequent cause of cancer-related death and is projected to become the second most frequent cause of cancer-related death by 2030. It is currently the deadliest form of cancer with a 5-year survival rate of less than 7%. Pancreatic tumors are highly resistant to chemo and radiotherapy, and unfortunately, PDAC is usually diagnosed at a very late stage once the subjects or patients have metastasis due to a lack of early symptoms and reliable imaging methods. So this is a terrible disease, and unfortunately, there are no reliable or effective treatments to treat pancreatic cancer, so any advances in understanding this disease are very much welcomed. So now let's move on to how cancer stem cells can be identified. This is an important point, especially when you want to study a disease or a tumor that is mediated by cancer stem cells. And for this webinar, what we will discuss is how can cancer stem cells be identified using flow cytometry. Now, the main method for identifying cancer stem cells has been and still is flow cytometry, which takes advantage of cell surface markers expressed on the cancer stem cell surface. The most common cancer stem cells markers used for pancreatic cancer are shown here in this diagram. For example, EpCAM, CD133, CD44, and CXCR4, common markers used to identify and isolate pancreatic cancer stem cells. Of course, isolating cancer stem cells is not trivial, and it's extremely difficult and oftentimes depends primarily on the use of antibodies that detect cell surface markers that are sometimes not exclusively expressed on cancer stem cells. For that reason, we actually set out to identify new cancer stem cell markers that could be identified primarily via flow cytometry, but independent of the use of antibodies. So in 2014, we published a study in Nature Methods describing a new cancer stem cell inherent marker that we termed autofluorescence. As you can see here, the publication. What we did is we used primary patient-derived xenograft cultures from PDAC samples, and we observed in these cultures very discrete green vesicles in our primary cultures, as you can see here in the first figure on the right. Interestingly, when these cells were cultured in conditions that enrich for cancer stem cells known as spheres, what we saw repeatedly was an increase in this autofluorescence. At the microscopic level, the autofluorescence was restricted to vesicles covered in the ATP transporter ABCG2, as you can see here on the left in this confocal image, using ABCG2 fused to M-cherry to detect its location and its localization within the cell. And the autofluorescence had a very specific excitation and emission spectra, which is very different than, for example, GFP. Most importantly, we could identify and isolate these cells using flow cytometry without the need for antibodies. And you can see here in these flow cytometric plots, in adherent cultures, we can detect a percentage of cells that are autofluorescent, and that percentage of cells increases when we put the cells in conditions that promote the enrichment of cancer stem cells. For example, putting these cells in conditions of sphere formation. Okay. And what we're also able to confirm is that these cancer stem cells, autofluorescent cancer stem cells, upon injection in nude mice, were always more tumorigenic than the non-autofluorescent cancer stem cells. ABCG2 accumulation on the membrane of these vesicles was only observed in autofluorescent positive cells. As you can see here on the right, autofluorescent positive cells that were sorted and autofluorescent negative cells that were sorted, we only saw the accumulation of ABCG2 in the vesicles in the cells that were autofluorescent. We later learned that the substance accumulated in these vesicles was actually nothing more than the naturally fluorescent vitamin riboflavin. Riboflavin naturally fluoresces at very high concentrations. Riboflavin can also pass membranes via the presence or via the aid of ABCG2 transporters. So it was not illogical that we would have riboflavin accumulation in vesicles that are coated in ABCG2. Now, since this publication, we've extended our analyses of autofluorescent cancer stem cells to more tumors as a means primarily of determining whether we can determine the cancer stem cell load in freshly resected tumors. So in collaboration with local hospitals in the area, we've analyzed 92 tumors to date, and we've been able to see that we've been able to detect autofluorescent cancer stem cells in a large number of these tumors. Interestingly, our ability to detect autofluorescent cancer stem cells is primarily restricted to tumors of the gastrointestinal tract, for example, colon, pancreas, stomach, rectum, gastric, and liver cancers. So this also highlights the fact that perhaps this cancer stem cell marker, autofluorescence, may be something unique to gastrointestinal tumors. Now, importantly, these cells that we've been able to identify in freshly resected tumors can also be isolated via flow cytometry and cultured in vitro, as shown here. This is subject 57, which is a rectum cancer. We were able to identify autofluorescent cancer stem cells, isolate these cells, and put these cells in culture. And you can see we were able to establish a culture, and these cultures maintained their autofluorescent vesicles. We're also able to inject these cells in small animal models, such as zebrafish. So shown here are zebrafish injected with autofluorescent positive cells and autofluorescent negative cells isolated from a colon cancer sample in the yolk sac, and then we were able to evaluate their distribution 144 hours post-injection. You can see that the autofluorescent negative cells that were injected were no longer present 144 hours post-injection, while the autofluorescent positive cells that we injected were still present within the zebrafish and had begin to migrate and expand and extend themselves within the zebrafish model. Now, while flow cytometry is a powerful tool for identifying cell populations, many factors should be considered, especially when working with rare subpopulations of cells such as cancer stem cells, and even more scarce populations such as circulating cancer stem cells in blood. For the purpose of this webinar, I would like to focus on the latter, which are circulating cancer stem cells in blood, but the same considerations that I'm going to discuss can be applied to cancer stem cells from tumor samples or also from cell lines. Now, when we talk about circulating cancer stem cells, we tend to lump this into the category of circulating tumor cells. And detecting circulating tumor cells has been a very challenging process. The existing paradigm states that circulating tumor cells can be isolated or detected based on the expression of cell surface markers, also filtration by size. Other techniques have used density separation to isolate and detect circulating tumor cells, and also, for example, white blood cells, a negative selection analysis. So basically eliminating the white blood cells and all the red blood cells and only remaining with the non-immune cells to isolate circulating tumor cells from blood samples. To do this, however, specialized platforms and instruments are needed. For example, the Verydex cell search system is one platform that is commonly used and FDA approved for certain types of cancers, and also other platforms exist. These exclusive and specialized platforms, however, are very cost prohibitive in some settings. Also, CTCs detected using these platforms use antibodies against, for example, EpCAM, which is an epithelial marker, or cytokeratins, which you could think are likely not present on cells that have undergone EMT or have assumed a more mesenchymal-like state. So maybe some of these antibodies that are exclusive to these platforms are not specific for the circulating tumor cell population. Also, these platforms are not designed to isolate or detect cell clumps or aggregates. So this also reduces our capacity to detect circulating tumor cells or circulating cancer stem cells that may travel as aggregates with support cells, which is something that the literature is now referring to as a very likely possibility for circulating tumor cells. That being said, obviously there is room for improvement. So therefore, we believe that a flow cytometry-based approach is the best option for identifying circulating tumor cells and circulating cancer stem cells. However, you can see here on the right, traditional hydrodynamic focusing, which is the traditional methodology used for flow cytometry, and using this hydrodynamic focusing, cells are not aligned in a continuous stream, resulting in many events being lost at the time of sample event acquisition. Now, this tends not to be problematic if the population you're looking for is present in very high percentages, but when you're looking for a very rare small population of cells, this can reduce your ability or the precision or your ability to detect this very small population. So to improve upon this, we have moved to acoustic focusing based cytometry using the Thermo Fisher Scientific Attune NXT cytometer that uses an acoustically focused sample stream to align cells for better precision and increased detection of very rare events, which is the type of cell population that we are looking for. Now, again, going back to the existing platforms, another inconvenience is that most platforms implement an initial step to eliminate immune cells. So we have to ask the question is that if circulating tumor cells and circulating cancer cells are traveling in circulation with support cells, maybe some of those support cells are actually immune cells. And if we implement an initial step to eliminate immune cells, we might be losing those circulating tumor cells or those circulating cancer stem cells. Also, some of the results from these platforms are inconclusive, sometimes variable, and the procedure is time-consuming. For example, detecting circulating tumor cells in pancreatic cancer patient samples is extremely challenging, and the Verydex system has not proven beneficial for detecting CTCs in PDAC patients. So therefore, I think what we need are more specific CTC markers to detect circulating tumor cells and circulating cancer stem cells, as well as newer, faster, and easier methods of detection. So regarding previous point number 4, where I mentioned that the elimination of immune cells from the sample may compromise our ability to detect circulating tumor cells or circulating cancer stem cells, as more studies are indicating that these types of circulating cells actually exist in circulation as aggregates with immune cells, what we've tried to do is take information from the literature and apply this to our platform. So there have been some very nice recent studies. For example, a study by Adams et al. Published in 2014 in PNAS that actually suggests that circulating tumor cells do circulate with immune cells. So in this study in particular, what the authors observed was that circulating tumor cells were actually fused to macrophages. And the cells in circulation expressed both epithelial cell markers, you can see here EpCAM positive, cytokeratin positive, and at the same time expressed immune cell markers, for example, CD45. So they were able to identify circulating tumor cells that were actually expressed markers also present on immune cells, and they suggested that these cells were actually circulating giant macrophages that had been fused with circulating tumor cells. Now, the question remains, is this a very radical concept? In fact, it's not. If we take a look at many of the studies performed in the Condoleezza lab at Albert Einstein University, they've been studying for years how macrophages are necessary for the intravasation of circulating tumor cells. Essentially, what they have shown is that macrophages promote tumor cell dissemination to secondary organs. They're absolutely necessary for the tumor cell to enter into circulation. Now, whether or not this is done via a fusion process or whether or not the macrophage just is along for the ride is still yet to be determined. In addition, we have shown that one way that macrophages may also promote circulation is actually to promote epithelial to mesenchymal transition, or EMT, via secreted factors. So using here in this example experiment, we've used patient-derived xenograft primary cultures, and we treated them with M2 polarized macrophage conditioned medium. And what we saw is a clear mesenchymal transition mediated by macrophage secreted factors as early as 8 days and as late as 25 days, treating our primary cultures with macrophage conditioned medium. So our working model is that TAMs, or tumor-associated macrophages that are present in the tumor, actually promote cancer stem cell dissemination by promoting epithelial to mesenchymal transition, or EMT, via secreted factors, or perhaps also by directly interacting with cancer stem cells or cancer cells to promote this fusion event. The latter is actually this idea of TAMs promoting cancer stem cell dissemination is actually biologically relevant in the case of pancreatic cancer, as CD163 positive macrophages, or TAMs, are the main immune cell infiltrate present in PDAC. You can see this here in this immunohistochemical staining of a patient of a pancreatic tumor. You can see here the massive infiltration of CD163 positive macrophages. We also see in blood samples, so here we have a sample from a healthy control. We can see a monocyte macrophage population of about 1.2%. However, when we look at the same macrophage monocyte population in blood from a metastatic pancreatic cancer sample, we see about a threefold increase in the percentage of these cells present in circulation. Now, if we look specifically at this population and we use a marker for M2 macrophages or TAMs, tumor-associated macrophages, here in this example we're using CD163, and via flow cytometry what we see is that we see 44.6% of the monocyte macrophages are CD163 positive, indicating that there's an increase in M2 or TAM-like macrophages in circulation, which are likely coming from the primary tumor. So the hypothesis is that macrophages promote cancer stem cell dissemination, intravasation, circulation, and facilitate their extravasation to secondary organs in order to form metastases. Now, this hypothesis was recently supported by a publication in 2017 by Clausen et al., where they observed fusion between circulating tumor cells in TAMs. As shown here, with a PDAC cell marker, which is ALDH1, and a macrophage marker, here they use CD68. And you can see that the cells that they were able to isolate from blood samples were both ALDH1 positive and CD68 positive, again indicating perhaps a fusion event between tumor-associated macrophages and circulating tumor cells. So we used basically the same approach, but applied it to a flow cytometry platform. So we use CD163 and our autofluorescence as our markers to detect double positive cells. And you can see here in sample 38, we were able to detect the double positive population. 3.24% of the cells were both positive for autofluorescence and positive for CD163. And when we isolated this population, we observed that these cells could form spheres. In vitro, these spheres also retain their autofluorescence phenotype. They were also able to form tumors in nude mice, metastasized to the liver following tailor-vein injection. And here shown on the right is a table showing that CD163 positive macrophages increase in PDAC samples compared to healthy samples. And we also see that an increase in PDAC in double positive cells, both positive for autofluorescence and positive for CD163 in our PDAC samples. So these data indicate that these TAM CTC fused cells could be used to develop a liquid biopsy assay. So to move this to more feasible screening liquid biopsy assay, we are now exclusively using acoustic-based cytometry, which allows for more precision and the ability to detect these very rare events when we compare this type of cytometry to more conventional cytometry or to the traditional hydrodynamic focusing cytometry. With acoustic cytometry, we're able to detect more events, sometimes anywhere from 3 to 5 times more events than we were able to detect using traditional hydrodynamic-based focusing. So here in this slide is an example of the detection limits of this approach. So in this experiment, what we used was fixed blood samples, and we spiked our fixed blood samples with pancreatic ductal adenocarcinomal cells. And we analyzed on the Attune NXT cytometer using the violet laser and in addition, together with the no wash, no lyse filter set to gate on nucleated cells without lysing the sample. So the no wash, no lyse filter set allows one to gate directly on nucleated cells. Therefore, the sample does not need to be lysed and the sample does not need to be washed. You can directly fix the sample or not fix the sample, add your antibodies to the blood, and detect your cell target without having to introduce extra variables such as lysing red blood cells, which can damage your cells as well, and the multiple number of washing steps that can reduce your ability to detect cells, and you can actually lose some of these very rare cells that you're trying to detect. So what you can see here within the population, we detected EpCAM positive PDAC cells with high efficiency and precision, demonstrating the utility of this approach. In the first column, you can see here, this is blood where no PDAC cells were introduced, and we have 0% detection of EpCAM positive PDAC cells. And then we did dilutions from 10 to the 6th down to 10 to the 3rd, and we were able to identify very well for when we should have detected 200,000 cells, we were able to detect 106,000 cells, 20,000 cells, 11,000 cells, 2,000 cells, 1,300 cells, and 200 cells we were able to detect. In theory, 200 cells we were able to detect 98 cells, showing very well the utility of this approach, the precision, and the efficiency using the Attune NXT cytometer. So our main hypothesis, therefore, is that circulating cancer stem cells enter into circulation accompanied by tumor-promoting macrophages. And we wanted to apply this methodology to primary samples, usually using CD163 as our marker for the tumor-associated macrophages. And our two markers for the cancer stem cells and tumor cells would be autofluorescence and EpCAM, respectively. Using the Attune NXT no wash, no lyse filter gating strategy, we can see here the nucleated cells which are present within the gate. Within that population, we can see very nicely the distribution of the immune cells, granulocytes, T cells, monocytes, etc. And then within this population, we can specifically detect the CD163 positive or tumor-associated macrophages using antibodies against CD163. As you can see here in this analysis, here we have a healthy sample, and within the CD163 positive population, we were not able to detect EpCAM positive cells nor autofluorescent cells in this healthy sample. However, if you look at the pancreatic cancer sample, we're able to detect very nicely within the CD163 positive population 2.18% of the cells that are EpCAM positive and 28.6% of the cells that are autofluorescent positive, indicating that our target population, this double positive population, is present in pancreatic cancer samples, but not in healthy samples. Now, if we look here in this analysis, we can see that, which is very interesting, those samples with the highest numbers of double positive cells, which are indicated here with the red bars, correlate very well to lower overall survival and lower progression-free survival. So those samples where we were able to detect high percentages of this double positive population, these samples correlated very well to low overall survival, which is basically what we're trying to look for, is a marker that will allow us to predict perhaps overall survival or progression-free survival. So we envision a cytometry-based liquid biopsy assay for the detection of circulating tumor cells that could be used for countless purposes in hospitals, with the main goal, however, of advancing personalized medicine options for cancer subjects. So we envision this technology being used, for example, for quantifying circulating tumor cells or circulating cancer stem cells before and after treatment to determine drug efficacy or chemotherapy efficacy. We also envision that this sort of platform could be used for monitoring circulating tumor cell or circulating cancer stem cell populations post-surgical intervention, and perhaps assessing the appearance of circulating tumor cells or circulating cancer stem cells as a predictive marker of cancer development or reappearance during remission. Of course, as with all platforms or all assays, sample preparation and choosing the right panel of markers is very important and needs to be empirically determined by each user. Our workflow is very specific to the type of tumor we work with and the type of cancer stem cells we work with and the tumor and the cancer stem cell markers that we have chosen. It is important that for sample preparation and choosing the cancer stem cell markers that each tumor may be different. What I will present here is our workflow and the requirements and the preparations that our samples undergo for our analyses. However, it's highly recommended that each user that develops a similar liquid biopsy assay empirically determine their conditions. So our samples come from the hospitals, and the samples are acquired from our patient population of interest. These samples, for example, blood samples are collected in various different types of blood collection tubes, for example, heparin tubes or EDTA tubes. And the nice thing about our platform is that you can choose whether or not to fix the cells or not fix the cells. If you choose to fix the cells, we have been using the transfix cellular antigen stabilizing reagent, which works very well for stabilizing the antigens on the cells, and the antigens are actually stable for up to 10 days at 4 degrees Celsius. Now, as I mentioned before, we take these samples and we run them through the Attune NXT cytometer. We do what we call a sample cleanup by using the violet laser in conjunction with the no wash, no lyse filter kit, which allows us to gate directly on the nucleated cells. And then this is the population of interest. So no need to lyse the cells, no need to wash. We can immediately go to the nucleated cells and determine whether or not ours are circulating tumor cells or circulating cancer stem cells are within the nucleated population. If we don't fix our cells, then what we tend to do is also add a dead cell exclusion marker so that we can exclude all dead cells and focus just on the live nucleated cells. Of course, you have to choose your antibodies of choice. For example, antibodies that can be used to directly mark your immune cells of interest. If TAMs or tumor-associated macrophages are of interest, we tend to use the Invitrogen antibodies against CD45 and CD163. However, other markers can be added, markers for myeloid cells, markers for T cells, etc. And of course, you have to choose which cancer stem cell markers are appropriate for your tumor type and for your conditions, and whether or not you are trying to identify circulating cancer stem cells or cancer stem cells directly from tumors. So, for example, you could use EpCAM to identify those epithelial cells in blood samples, pan-cytokeratin, for example, and of course, we also include our autofluorescence marker. And once the staining has been performed, we can run these samples very efficiently and thoroughly through the Attune NXT cytometer. We do a multicolor analysis and a quantification. You can actually quantify the number of events or the number of CTCs or circulating cancer stem cells using a count-bright absolute counting beats. And in the end, obviously including your healthy samples together with your experimental samples will allow you to gain confidence in your results and to compare the signal you receive from the experimental sample compared to the healthy sample. And with that, I would like to thank you all for your attention. I would be more than happy to further explain any points in the presentation or to answer any questions.
Thank you, Dr. Sainz, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Sainz will answer as many questions as time permits. The first question is, can you combine the autofluorescence with well-known cell surface markers to increase the ability to detect cancer stem cells from tumors or from blood?
So you can definitely combine the autofluorescence marker with any marker of interest. And the literature, as you read in the literature, I think just about every month a new cancer stem cell marker is being identified. So the ease of the autofluorescence is that it is antibody independent, which then allows you to include more antibodies without having issues of isotype controls, etc. The only limitation, however, is your channel for green fluorescence has been occupied by the autofluorescence. But you can definitely combine other markers based on the number of lasers you have, especially if you use the violet laser, then you have an even larger gambit of antibodies that can be included.
Thank you. Why do cancer stem cells accumulate riboflavin in these vesicles, and do you think there is a biological benefit?
That's an excellent question. Why they accumulate the riboflavin at the biological level or at the mechanistic level? We know they accumulate it because they overexpress ABCG2 on the surface of these vesicles. So naturally, riboflavin is a ligand for the ABCG transporter. So it will pass through the membranes that have this transporter, and the fact that the vesicles overexpress them, and the natural result is its accumulation within the vesicles. Now, whether or not this accumulation of riboflavin has any sort of biological benefit, that I think we still do not know. Obviously, riboflavin has many biological functions. It's a precursor for the flavoproteins, so it may be that the cancer stem cells are accumulating riboflavin to ensure the production or the availability of flavoproteins, which function in many different biological processes. But I think that question is still open and unanswered.
Thank you for that. The next one is there one universal marker that could detect all the cancer stem cell subpopulations across all tumor entities?
That would be fantastic, and I wish. It is nice that the autofluorescence biomarker, it seems to be a very good marker for identifying cancer stem cells of gastrointestinal origins. The only thing I would be cautious to say is that do there exist cancer stem cells that are not autofluorescent positive? Perhaps, and most likely. Do there exist cancer stem cells that express markers that we have yet to identify? Probably. So I think if we limit ourselves to one marker, we are just getting a glimpse of the cancer stem cell population that expresses that marker. And we also should always keep in mind that there are other cancer stem cell populations that probably express other markers that if we don't use those markers, we're losing that information as well.
Thank you. Looks like we have time for one more question. What do you recommend for number of cells required for analysis? So
I can answer in two parts, from primary tumors and from blood samples. From primary tumors, I would suggest at least, if it's possible, anywhere from 5 to 10 million cells is a good amount of cells to do an analysis for cancer stem cell percentages. We have to keep in mind that depending on the tumor type that you're studying, the cancer stem cell population can be anywhere from a 0.5% to 5% to 10%. So I think that needs to, based on those percentages, you can extrapolate the number of cells that you would need. For merely detecting the cells by flow cytometry, you can obviously reduce those numbers of cells. And obviously with the Attune NXT cytometer, you gain greater confidence and precision because you're able to capture each and every event. If you're using traditional traditional cytometry, you might want to increase those numbers. From blood samples, a good starting volume is about 5 mLs of blood, which can be used and marked and analyzed as mentioned in this presentation.
I would like to once again thank Dr. Sainz for his presentation. Do you have any final comments?
No, I would like to thank the opportunity to present our work and the experiments and the research that we're doing in our laboratory. Of course, thank also Thermo Fisher Scientific for this opportunity and you all for your attention.
Thank you for that. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through June 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.